Welcome to this, I think it's our 10th Monday session. And before we get started, I'd just like to thank those of you who have already sent me in your experiences of the course. I've got a couple of these already. It would be lovely if any of the other participants would like to send me just a few lines about things you've enjoyed about the course or things you would like to see in the future. That'd be much appreciated. So you can send that to my email address, or you could send them to Hui, and he can pass them on to me later on. Then just to remind you that next week, the last session is with Sean Watwell, and it's going to be an application of risk assessment at the Sudbury site in Canada. So we're looking forward to that one. But today we have Ian Oliver back again from Keele University. And he's going to follow on from his lecture last week on the principles of ecotoxicology. And today he's going to be telling you about how ecotoxicology is used in chemical risk assessment. Now, if you missed the first part, you can catch up online, but I hope you enjoy the second part today.
Okay, folks, I hope you can hear me and that you can see the screen this time. And we will crack on. Right, we are talking about terrestrial ecotoxicology and how that can be used in chemical risk assessment when protecting the environment. We'll be talking about the chemical risk assessment process for the environment, how we can do some initial screening of a situation, and then how can we derive predicted no-effect concentrations from ecotoxicology data. We'll also go through how data can be evaluated and determined whether it should be used or not used in that process. We'll also talk about how to combine different data sets. And then we will finish with a part specifically on metals, as many of us are very interested in metals and bioavailability. And we will look at how terrestrial biotic ligand models, or BLMs, are now being used in environmental risk assessment and regulation. But first, I thought I would do a one-slide recap for those who missed last week's session, just so some of the terms are familiar to everyone. We talked about what ecotoxicology is and how it aims to determine the concentrations below which no significant harm is anticipated. And we determined those concentrations by exposing organisms to varying concentrations and then assessing the level of inhibition. To do that, there are a few approaches, including exposing the organisms to different concentrations and then determining the highest concentration that causes no observed effects, or NOEX. The more modern approach and the preferred approach is to have many more concentrations tried and then to plot them on the dose response curve and interpolate between those points, and from that interpolation determine the EC10, so the effect concentration causing 10% inhibition, or the EC50, the concentration causing a 50% inhibition. Whichever way we choose, we can produce these ecotoxicology endpoints. And if we combine endpoints from different species, from different trophic levels and different soil types, then we can derive our predicted no-effect concentrations. So if we're going to do a chemical risk assessment, let's step back and think about what is in the person's mind. If you are in charge of a site and you need to remediate it, or you are regulating the use of a chemical, you have to ask yourself, does chemical X cause a problem at concentration Y milligrams per kilogram? Does it pose a risk? The first step is to find out whether there is a recognized environmental quality standard, or EQS, or an eco-SSL, an ecological soil screening level. If there is, then your job is very easy. You can just compare your site measured concentration to that EQS and determine whether there is a risk. Very often, though, there is not a valid or recognized EQS or ECO-SSL, So you have to derive one yourself. We can do that quite quickly and easily in a screening PNEC approach, and then we can compare and assess the risk. Then if necessary, we can go on to derive a full predicted no effect concentration using our ecotoxicology data. So that's what today's lecture will focus on. So to do that screening step, that screening PNEC, or sometimes called a first tier risk assessment, we can use what's called the equilibrium partitioning approach. This is for organic chemicals, so not for metals at this point. We'll come back to metals at the end of the lecture. But for organic chemicals, we can estimate a PNEC for soil using a PNEC that has previously been derived for water. Bear in mind that for most chemicals, there is an abundance of information about aquatic toxicology, so we can use that to estimate a soil PNEC using that PNEC derived for water, or an environmental quality standard derived for water, and the partitioning coefficient, or KD. And if you remember back to the first lecture in this series, Professor Sauvé went through partitioning coefficients and why it's important in soil situations. Here, we get to use it again. It is simply a concentration in the solid, so in the soil, divided by the concentration in water, so the pore water in our case. So in order to derive our screening predicted no effect concentration for our soil, we take our KD, our equilibrium partition coefficient, and multiply it by the PNEC for water, or the environmental quality standard for water, and we get our value. And then we can calculate a risk quotient, or RQ. Here we take the predicted environmental concentration in our soil, or if you have measured the concentration at your site, the measured concentration for your soil, and we divide it by our screening PNEC. Now, if our risk quotient is a lot smaller than 1, so we're talking orders of magnitude smaller than 1, then straight away you know that there is very little risk, and there is very unlikely to be a problem. But if your risk quotient is closer to 1, or even above 1, then you need to go on and do a full risk assessment and derive a proper predicted no effect concentration. But let's have an example of a screening PNEC. This is the example for hexachlorobenzene using the equilibrium partitioning approach. Its KD is 726, that's litres per kilogram, and previously its PNEC in water has been calculated as 0.0037 milligrams per litre. So we can use those two values, the KD and the PNEC in water, to calculate a PNEC for soil. We simply multiply 726 by 0.0037 and we get 2.7 milligrams per kilogram as our PNEC for soil. So now we would do our risk quotient and look as to whether it is much smaller than 1 or not, and decide whether there's no risk or whether we need to go to the next step and derive a proper PNEC. But that relies on you being able to find the KD for your chemical. So I hear you thinking, what if I cannot find the KD for my particular chemical of interest? If you look in a reference textbook or online, various databases available, you don't always find the KD for the chemical that you need to assess. But what you might find is the KOC. Now, the KOC is the organic carbon normalized partition coefficient. So that's determined by taking the KD and dividing by the fraction of organic carbon in the sample. So if you go onto an online database and you find the KOC, then you can work out the KD for your soil by either using the fraction of organic carbon in your soil, or if you want to make it more widely applicable, you can put in a general value, say 3% or 4%, which would be a typical organic carbon concentration in an agricultural soil. And then you can calculate your PNEC using that approach. But what if you can't find either the KD or the KOC? I've been in this situation, and you might be if you are a regulator using or trying to regulate the use of new chemicals or chemicals that are not usually used in that setting, or you might be doing a PhD on a new chemical and you want to calculate these values. If you can't find KOC or KD, then you can estimate your KOC from the KOW. Now, I know I am throwing a lot of Ks around there, but do bear with me. The KOW is the partition coefficient between octanol and water. That's what the OW stands for, octanol and water. And De Toro et al. In 1991 derived this regression equation from which we can estimate the KOC from the KOW. Now, to find the KOW, you can look it up in a textbook or online, or you can look up the solubilities in octanol and water for your chemical, and you can simply calculate the KOW. Once you have that, you've got your KOC. From that, you can get your KD. You can go all the way back and calculate your screening PNEC. And there was some further work done by Sabelich et al. In 95, which refined some of these equations so you can get regression equations for specific sets of organic chemicals. All right, so that's the equilibrium partitioning approach. It's useful as a screening step, but it does rely on lots of assumptions, including that concentrations in pore water are at equilibrium with that in the solids, and that concentrations in one phase can be predicted from another phase. It also relies on the sensitivity of soil organisms being similar to aquatic organisms. And finally, that effects in the soil can be predicted from effects in the water and using the partitioning coefficient. They're all quite robust and reasonable assumptions, but it does ignore ingestion as a route of exposure. So for organisms such as earthworms that actually ingest the soil and process it internally, it doesn't capture those extra sources of exposure. And that is why when you're doing an equilibrium partitioning approach, it really is only a screening PNEC, and you want to be making sure that your risk quotient is a lot smaller than 1. If it's anywhere near 1 or above 1, you need to go to the next step to properly assess the risk. So if we're going to do that, then we need to derive our PNEC from soil ecotoxicological data. Now, to do that, we need valuable and valid and reliable ecotox endpoints. So in order to do that, we can combine all the data that we can find, or all the data that we have produced ourselves, and then screen it for reliability. You will sometimes hear terms used such as Klimich codes. Klimich was an author that was working in aquatic risk assessment and came up with this idea of screening values for reliability. So sometimes you'll hear Klimich codes used. But really, we're just establishing a reliability index. So we can go through, look at all of the endpoints that we have found and identify them as being very reliable or not reliable. And we apply this index RI 1 to 4, with RI 1 being completely valuable and useful results, and RI 4 being not very useful at all. In order to rank as reliability index number 1, you would look to see behind the data, how was it derived? Did they use a standard test protocol? For example, did they use an ISO or an OECD test protocol? If they have done that, then you know exactly the conditions that the tests were conducted under. You know the temperature, the pH, all the information is there. You know the length of time that the exposure was done for, so you can be completely confident that the data point is valid. Use that one, no problem. But if they have not used a standard protocol, but there's loads of information in the report or in the paper, for example, they've included soil pH, the organic carbon percentage, the texture, they've used controls and lots of dose levels, they've described how they've mixed the chemical into the soil, they've explained how they have allowed it to equilibrate, they've given you information about the species being tested, and they've presented the data in a way that statistics can be done on it. That's still pretty darn reliable. So you would call that reliability index number 2, and you would use that data point also. Other studies that might not have a standard protocol and they might have some details missing, you might call that reliability index 3. And then if you've only got a data point with no detail or very limited detail, you might call that reliability index 4. And you would do that for all of the data points that you have. And you would use all the ones that you have rated RI1 and RI2. And depending how many you've got, you would use or not use RI3. You would exclude R4s because they're not reliable. But if you just don't have enough data, then you would fall back and use the RI3s also. So once we've got that data, we've screened it for reliability, and now we want to derive our PNECs. There are two different approaches that are commonly used, and it depends on the amount of available valid data that you have. The first is called the deterministic method, sometimes called the assessment factor or AF method. And the second is a probabilistic approach, which uses a species sensitivity distribution or SSD. The way to choose is quite simple. If you only have a few data points, then you need to use the deterministic approach. But if you have many data points, then you can use the more powerful probabilistic approach. So let's have a look at the deterministic approach, the AF approach. Here we look through all of the data points that we have, we identify the lowest valid endpoint, and we divide by an assessment factor. So our PNEC is our lowest valid EC10 or our NOEC, and you divide by the assessment factor. So let's say in all of the data you have found for a particular chemical, the lowest EC10 was 20 milligrams per kilogram, then you would divide that value by some assessment factor, in this example 100, to determine your PNEC as 0.2 milligrams per kilogram. Then you can do your risk quotient assessment and see whether the predicted environmental concentration in the soil is smaller than this predicted no effect concentration. If your risk quotient is less than 1, no problem, no risk, everything is fine. You have remediated your soil to a level that's fine. But how do we determine what the assessment factor should be? How should we choose our assessment factor? That is determined by how much uncertainty is associated with our data. If we have low uncertainty, then we can use a low assessment factor. If we have high uncertainty, then we need to use a higher assessment factor. So what is this uncertainty I'm talking about? Well, think about what we're doing with ecotoxicology. We're trying to find values that cause no significant harm to the whole ecosystem. But we're starting off with data for just a few species, and we've got to extrapolate that out to protect whole ecosystems. Similarly, we might have data from laboratory tests, but we want to protect the situation in the field. We might have short-term tests, and once the chemicals are out there, they're out there forever, so we have to extrapolate from short to longer periods. And we also have to extrapolate from the limited range of conditions that our tests are done in to all conditions experienced in the field. So these are the areas of uncertainty that come into it. And so we capture that in our assessment factor that we use. If we're going to derive a predicted no effect concentration using this method, then we want to make sure that our data covers primary producers, consumers, and decomposers. That way we're covering more of the trophic levels. And we have a look to see what our data covers. If we have really limited data and we've only found a short-term toxicity test, let's say for a new chemical, so we've got one short-term test for earthworms, and that's the only data point we have, then our assessment factor has to be really high. So we use 1000. And if that is the case, then we should also do a PNEC calculated by the equilibrium partitioning approach so that we can then compare and use the more conservative value. But if we have a bit more data, let's say we have a NOEC or an EC10 for a long-term test, for example, plants, now we don't have so much uncertainty in terms of extrapolating from short to long term. So then we can drop that assessment factor down a bit, bring it down to 100. If we have more data, if we have NOECs or EC10 for long-term tests for two different trophic levels, let's say for plants and for earthworms, then the uncertainty is dropped down further and we can bring down the assessment factor to 50. And then again, let's say we have long-term data covering 3 species and 3 trophic levels. So now we might have earthworms, plants, and microbes data for all of those 3 trophic levels. Now we have more certainty across a wider scope of the ecosystem, so we can drop that assessment factor down to 10. But if we've got lots of data, we can do species sensitivity distribution approach. So that is the probabilistic method or SSD method. This is a preferable approach because it allows multiple organisms to be viewed together. And therefore, the data set is more reflective of the whole ecosystem. But it does require more data points. It's a statistical approach, and therefore the statistical assumptions have to be met. And therefore, we really need at least 10 data points, preferably 15. And we really do want to cover the primary producers, the consumers, and the decomposers in the environment. So the way to go about that is to, again, collate all of the valid EC10s and NOECs that we have found. So if we've gone through, found all the data points, we've done a reliability index assessment, all of our RI 1s and 2s, maybe our 3s, then we rank them from lowest concentration to highest concentration. And then we can transform them logarithmically. Then we plot that out in a cumulative distribution of dose versus effective percent or affected fraction of test organisms. And we come up with this kind of graph here. So here we have our cumulative distribution function, or our species sensitivity distribution. We have on the y-axis the potentially affected fraction, or the proportion of organisms impacted. And on the x-axis, we have the log 10 toxicity data. So each of these data points represents an organism type or a species, an EC10 value or a NOEC value. That's been log transformed. And then we calculate the HC5. So that is the concentration that is protective of 95% of all organisms in all situations. Or if you like, it's the concentration that is hazardous to 5% of cases, the HC5. And then we can use that data point to calculate our PNEC. Here is where the fudge factor comes in. We take that HC5 and we tend to divide by some kind of safety factor, which is a bit of an art rather than a science, but it's selected based on how much data we have. If we have hundreds of data points, then our uncertainty is very low, and so we would use a very low safety factor. If we've got data from laboratories and from soil that's been contaminated in the field and field aged, then that takes away some of the uncertainty of going from lab to field. Therefore, again, we can drop that safety factor. So if we've got field data, lots of data covering many organisms, many soil types, then we might just use a safety factor of 1 and use the HC5 as our PNEC. But if we haven't got much data at all, let's say we've only got the bare minimum to satisfy the assumptions of this SSD approach, then we might apply the safety factor of 5. And if we've got data volume somewhere in the middle of those things, then maybe we'll use a safety factor of 2 or 3. And it is an art, and regulators get together and argue about which safety factor to use, and then it's just a weight of evidence approach, and the best argument wins, basically. And you use that to determine your PNEC. If you wish to do this yourself, there is some very good free software available. The one I use is ETX2. You can just download it and use it on your own computers. It's very nice because it calculates the HC5 and it calculates the confidence intervals around the HC5. And it generates lots of different graphs for you to use if you're doing a presentation or wanting to put these into a report. And I've used these for client reports previously, and it's very good to show things visually. Do note, though, that it tabulates the data in scientific notation, so don't let that confuse you. So 2.4 E plus 1 instead of 24, for example. There are other software and formats available, including if you go online and look for Mosaic, it will also do a species sensitivity distribution for you. It's quite a good platform online. When we're combining data, we have to bear in mind that soils are really variable. Water is far less variable than soil. Soil has pH that typically ranges anywhere from 3 up to 9, and it has different organic matter levels, different cation exchange capacities, and of course different textures, different clay levels, which will completely change the bioavailability of chemicals. So if we're going to combine our soils data to derive a single PNEC, we have to think about how we're going to do that and what is the best way to do it. So I thought I would show you some of the things that I considered and how it is done so that you can do it yourself. The first option is just to use all the data as it comes. Just use it as it is, throw it in, calculate your HC5 and your PNEC. The justification for doing it that way is that if your data set is broad enough, then it will include ecotox assays conducted under what we might call sensitive conditions. So sandy soils with no organic carbon in them, that's a very sensitive soil. If your data set includes those kinds of soils, as well as clay-rich soils, and if your data set includes pHs from 3 to 9, then you can be pretty confident that your HC5 is predicting 95% of all conditions, including the sensitive ones. So you can just use your data, calculate your HC5, calculate your PNEC, and just say it is protective, and there it is. So that's one way to do it. But you can get a bit more sophisticated also, and you can normalize your endpoints to a key parameter. The one that is often used is organic matter. You can go through and normalize all of your endpoints, all of your EC10s, or all of your NOECs, and calculate them on an even and fair organic matter concentration. The justification for that is that organic matter highly influences the bioavailability and toxicity of chemicals in soils. So you can go through and normalize based on that. And here is a calculation of how to do that. You have your NOEC or your LC50 or your EC10 that you have found online or found in a journal article or you've developed yourself, and you want to convert that to a standard or standardized NOEC or EC10. To do that, we simply multiply by the fraction of organic matter that you want to standardize to, divided by the organic matter in your own soil. Typically, we would use a soil organic matter fraction of 3.4%. So if your soil was 5% organic matter, you would multiply your NOEC or your E-sensitive organism type, which is quite often useful to know. If you wish to learn more about this, then you can look up the rulebook. This is the rulebook for us in Europe. This is the technical guidance document on risk assessment. This is part 2. It has about 300 pages just in part 2, so it's quite the heavy tome. But all of these technical guidance on risk assessment were transposed into our chemical regulation REACH program, as you see here on the right. Every chemical that's used in Europe needs to be registered and evaluated under the REACH guidelines. And so those REACH guidelines now specify the guidance for how risk assessment is done. And there are parallels for other jurisdictions too. And indeed, if you want to look those up and you want to read more into this area, some useful textbooks are shown here. The one on the left by Van Leeuwen is very good. It explains lots about ecotoxicology. It also talks about chemical risk assessment, and it covers the approach in North America and the approach in Europe, and it compares the two. They are fairly similar, but it's always useful to have the specifics pointed out for you. The textbook on the right by Newman is another very good textbook. Again, it covers fundamentals of ecotoxicology, and it also goes into risk assessment. So if your library has any of these, go and have a read. They are really good books. I don't get any kind of commission for sales of these, so don't worry about that. Just go ahead and enjoy them. Right, so let's move on to metals. Let's finish with metals, given that we are mostly interested in metals, at least the people that I have spoken to in the IIES have quite a focus on metals. Soil ecotoxicology for metals is much more established than for many other kinds of contaminants and pollutants. And so the science is the most advanced for metals compared to other contaminant groups. There's been a whole host of very large international projects, including ones that were funded by metals producers. I did a project for ILSRO, that's the International Lead and Zinc Organization, a few years ago. Similarly, when I was at CSIRO, we did work for the Nickel Producers Environmental Risk Association and the International Copper Institute. So the big metal production agencies, they have an interest in this, and they quite often fund science in this area. And they've been responsible for getting a lot of this work done, which is to their credit. And in case you wish to look up some work by some of the big shots in this field, I've listed a few names there that you can pursue at your leisure. People like McLaughlin Lomby in Australia, who I used to work with, Yibing Ma. These are just the ones that I have followed over the years. apologies if your favorite isn't listed among them, there are others, but these are some of the heavy hitters and some of the authors of publications that are really worth looking up. So I said that metals are the most developed in this area of ecotoxicology. Because of this, we can start to bring in bioavailability much more into the assessment of risk. Now, for metals, as Professor Sauvé pointed out in his first lecture in this series, the free ion activity is what can help to predict the bioavailability and the toxicity of metals. And so we can turn that around and predict the bioavailability from the properties that influence the free ion activity, so things like pH and organic matter. And Professor Sauvé, back in 1998, did some excellent work on this to derive regression equations for predicting the free ion activities of copper and lead in soils. Now, I cited these 26 times in my PhD. I went and counted up. So this was very influential, and work has built on this over the intervening periods. Now, because ecotoxicology and bioavailability are inextricably linked, all of that work dating back from the 90s, even back to the 80s, all the way up to now, is now being pulled together into the development of what we call terrestrial biotic ligand models, or BLMs. These are some of the BLMs that are now available. This is for deriving a site-specific PNIC for whatever metals you are trying to look at. They are being increasingly used in environmental regulation. Indeed, the UK is bringing in these soil PNIC screening terrestrial biotic ligand models for calculating site-specific PNICs. And so they're becoming more and more used. They are currently available for copper, zinc, nickel, lead, and cadmium, but more are being produced all the time by various environmental consultants. This particular one that I'm about to show you was produced by Koen Oort's group in Belgium. Here, you put in the metal that you want to calculate a PNIC for. So in this case, I've done it for lead. If you know it, you can put in a background concentration. You can tell what kind of soil you have. So here I've set a sandy soil. And then you hit go to output, and it will calculate for you a PNIC for lead. So it will calculate a PNIC, and it will calculate a risk ratio for you as well. So as you can see, for the situation in a sandy soil that I put in, it's come up with a risk ratio much less than 1. So for this soil, there wouldn't be a problem. And note that it gives you the PNIC on total metal and also the added approach. You can do it for the other metals as well. You can do it for nickel as an example. And if you scroll down or look at the bottom of the screen, you'll see that for nickel, as for zinc and copper, the models are a bit more sophisticated, and they can bring in much more specifics to do with soil pH and organic matter content and percentage clay. So if you have that data for the soil that you're trying to examine, you can plug that into the BLM, and it will calculate for you, again, a PNIC for nickel, and it will give you a risk ratio. So in this scenario, you can see that the risk ratio, the RCR in column 3, is much greater than 1. So here we would see that we have a potential toxicity problem. You can also do it for multi-sites or multiple soil types. So let's say you wanted to examine how the PNIC for your metal varies with soil pH. So here, if you squint and look closely, you'll see that the organic matter and the clay content I have kept constant, but the soil pH I have varied between 4 and 9. And as you can see, the BLM calculates differing PNICs based on how the bioavailability and therefore the toxicity is affected by that pH variation. So here you can see that the PNIC goes from 43.7 if the soil pH was 4, all the way up to 58.1 if the soil pH was 8 or above. So you can see you can factor in things like organic matter, clay, and pH to your PNIC calculation using these biotic ligand models. And the data for this are based on huge databases. This is just one little snapshot to give you an idea. So all of those ecotoxicology tests that have been done on metals over the years are now available in databases, and that data is used to derive regression equations upon which we can use to calculate site-specific PNECs. That's pretty much it, folks. So in this lecture, we have covered risk assessment. We've talked about how we can do an initial screening step. We've talked about how to derive PNECs using ecotox data. We've talked about evaluating our data for reliability. We've talked about how to combine data sets using different soils. And then finally, we finished by talking about the special case of metals and how bioavailability can be incorporated into PNECs using things like the terrestrial biotic ligand models. That's it. I think we'll do some questions. I'll unplug my headphones and hand over to Dr. Graham.
Thank you very much, Ian. That's a really interesting presentation today. Who would like to start off with some questions?
Well, I've got a question for you, Ian. Models are nice because they spit out a number. And of course, regulators like to have a number spit out at them. But if the final table that you were showing there with the metal BLMs, you noticed over the pH range that you chose, it gave values to 1 decimal place of a PNEC, and varied from like 45.7 or something to 55, something like that. Realistically, are those numbers over that entire pH range any different? And do you really think that values to 1 decimal place are reasonable?
Values to 1 decimal place, regulators would not use. They would just use it to, they would have it rounded off. That was probably the least sensitive example I could have picked. If you varied the organic matter and the clay content more, you would get a much wider variation in addition to that variation seen by the pH. What regulators tend to do is they say your waste material cannot be applied to anything below 6 if pH 6, if your pH is known to have a big impact on the availability of the metal, for example. So they would have a lower limit to what you're allowed to apply, and they would put in their own safety factor typically. But no, they don't use those decimal points. But that's what the model puts out for us. I suppose also they use them these days as a screening step. So if the measured or estimated concentration is a lot higher than that, then they go and do a full risk assessment. If it's a lot lower than that, then they say it's fine. But they use that as a first screening step, and then if necessary, they go on and do greater evaluations.
I think maybe you should stop share.
Oh, there we go. Oh, there we go.
Yes, now we can see this. Yeah, that's good.
Bring up chat. Okay.
Yeah, because there may be some chat questions. Yep.
Good one. Did that answer the question, Doug?
Yeah, I think, my point is that sometimes models tend to lure us into thinking that that a number is perhaps more significant than it might be. So I think your answer was right. As long as it's used in a general way, that's fine.
Yeah. And a number is very reassuring because they can just go, is it bigger or is it smaller?
Yes, yes. Do we have more questions?
Yes, I have a question, or 2 questions. One is the role of speciation. From Margaret, we heard that for chromium, it really depends on the speciation of the metal. So in these models, to what extent is speciation considered? And second question is, if you have several metals, usually you have a contamination not only from one metal, but a combination of several metals. So how do these models cope with that problem?
Yeah, not very well is basically the answer. For things like chromium, they don't do speciation because they don't have the data or the regressions available. So to my knowledge, chromium, there is no terrestrial biotic ligand model available because they're too scared to go ahead with the limited data that they have. They need more research done. So that's why there's only these models available for maybe 5 metals at the moment, because they're fairly well understood. And for things like lead, speciation doesn't matter so much. It's either there in the poor water and it's toxic or it's not. So yeah, they can't capture that. That's one thing. And in terms of the multiple contamination, yes, that is an issue, as you say. Old gasworks sites or old mining sites, they're contaminated with loads of different things. And if you have additive effects or synergistic effects, then just finding out that the concentration of zinc is below a threshold, it doesn't really cut the mustard. So what they do is just look at it individually and say, does it meet all individually? And if so, they give the thumbs up, because there isn't the additive models yet. They are coming and people are doing research on them, but they are years away from being used in any kind of regulatory sense, because the regulators have to be certain. They have to have very little uncertainty. Otherwise, their regulation just doesn't have a legal leg to stand on, and they'll get sued by anyone that they try to shut down.
Okay, I see a question coming in chat.
Michael?
Yes, from Michael and the group. How receptive are regulators to changes and updates in the models? And second question, do they tend to use outdated systems or update them regularly in line with new results?
That's a very good question. The regulators quite often fund environmental consultants to carry out this work and do calculations and to develop biotic ligand models. But then, as I was just answering previously, they don't commit to them until they have high certainty, or if they have high confidence. And for us in the UK, that means, do the Germans accept it? Do the Swiss accept it? If everybody else is accepting it, then our regulators will accept it. If the US EPA is accepting it, then maybe we'll accept it. So because of that process, that takes about 10 years. So some of these BLMs have been around for 10 years, but until everyone gets confident using them, the regulators aren't that quick to take them up. But once they have them, then they use them usually for some kind of cycle, and then the next one, updated models will come in. But because they have to regulate on things over a period of years, they can't change things every year. They have to bring in regulatory changes. They have to advertise them. They have to have consultation on them. So the updates tend to happen quite slowly as well. So they're always a bit behind the science, simply because they have to factor in all those practicalities.
Okay, hopefully that answers the chat question. Yep, there's a thanks. Okay, well maybe I can ask a question and just take you back to the beginning of your lecture, when you were talking about, probably mainly for organic contaminants, the use of the KD approach. Does this really only apply to systems that are like at low contamination level? Are there any problems about applying that approach when you move to higher organic contaminant concentrations?
It's a screening step. So you do that calculation, and it gives you a ballpark approximate value of what it's likely to be like in the soil and whether it's going to be toxic or not. And so if it's anywhere near, if the risk quotient is anywhere near 1, then you know that you've got a potential risk situation, and therefore you have to go ahead and do the full scale phenolic derivation, which sort of answers your question in reverse. If the concentration is really high, it's very likely to be well above a risk quotient of 1, and therefore you've just proven that you need to do the full phenolic derivation.
Yeah. Okay. That's fine. And so if you outlined then there are two different approaches, the deterministic and the probabilistic methods. Are both of these being used interchangeably, or like would the regulator use both of them, or would they only be confident if they had much more data?
No, the regulators have to make a decision. If someone's going to use a chemical at a site and they want to use it, and it's a legally
registered chemical, then the regulator has to decide is it safe or not to use. And so if there's not enough data, they still have to make a decision.
Yeah.
And so if there's not enough data for the probabilistic species sensitivity distribution approach, then they have to use the deterministic approach, and they do.
Yeah. So there would be a conversation between the regulator and the company, you know, like for a small scale discharge or something like that.
The regulator would assess the data, see how much is available, and depending on how much is available, they would use the appropriate approach, and then they would go to the company and say, your concentration is too high, you can't do that, or not.
Mm-hmm.
And they would explain to them how they derive their number,
Yeah.
but it's on the regulator to decide what's the appropriate way given the amount of data available.
Okay. Do we have any more questions? Okay, well, I think we're just about done then. So thank you very much, Ian. I've learned a lot today. This is not an area that I knew very much about. I think I'm going to be going and looking up some of the references that you've been suggesting, and some of the texts as well. So thanks very much, everyone, for joining in the discussion today. And I look forward to seeing you again next Monday for our last session with Sean Watchmo. But for now, I think we'll say goodbye, and thank you very much, Ian.
Thanks, everyone.